everybody to this afternoon's session on it's a talk. We call this MNBF Talk, the Micro Nano Bio Fluidics Work Group's main colloquium or invited talk. Today we have the pleasure of having Professor Balpreet Singh Ahluwalia from the University of Tromsø, the Arctic University of Norway. He has very kindly consented to present this colloquium style talk on photonic-chip based super resolution optical microscopy. Indeed, this would be a topic of considerable interest under the auspices of this group on micro, nano, and biofluidics. So we are a confluence of fluidics, then biology, and bringing in together the science and technology that are associated with it. It gives me great... Pardon?
This is Vishal. Can you hear me, Shiva?
Yeah, Vishal, we'll come back to you.
Yes.
Yeah, we can hear you now.
You are able to hear me.
Yes. Thank you. Yeah. Okay, so yeah, we have several colleagues from IIT Madras and elsewhere joining the Zoom meeting. We also have a live YouTube stream. And to put this in context, this talk series and this talk itself is being organized by the Micro Nano Biofluidics work group. This work group is also called a Prospective Center of Excellence. This is funded under the aegis of the Institute of Eminence scheme of the Ministry of Education of Government of India. IIT Madras is one of the cluster of institutes of excellence, along with other IITs and IISc and the Institutes of National Eminence. Our group primarily focuses on doing and developing cutting-edge research in micro nano biofluidics and in developing disruptive technologies by exploiting material and energy transport on micro and nano scale on aspects of biology, then energy sciences and beyond. We are really a very interdisciplinary group, and the group essentially is constituted by about 10 faculty members and research teams. Our research plans are in areas of interfacial microfluidics, nanofluidics, and lab-on-chip technologies. And especially in that context, today's talk is going to present a relatively new dimension in this talk series. This is about the 7th talk in this series. It deliberately has a leaning towards photonics. The interest of this, our group also lies in understanding the fundamental science in fluid streams, interfaces, droplets, particles, cells, and energy transport. We are equally both committed to doing research on fundamental principles as well as developing techniques and technology in this area of research with particular applications to healthcare, energy, and water. And we are indeed today very glad that an eminent young scientist like Balpreet is joining us to make this colloquium live and online. To tell you a little bit about Balpreet, and in the interest of not taking too much time, Balpreet's career began with his PhD in the Nanyang Technical University, and he then soon took up a postdoctoral position at his current affiliation, the Arctic University of Norway in Tromsø, and he currently holds eminent visiting positions, including the one at the University Hospital in Stockholm in Sweden, the Karolinska University Hospital. He has been extremely successful, not just in his research in basic sciences and technology in the lab, where he has published over 70 journal articles and contributed to several conference proceedings, and even at this fairly young stage has an H index in over 20. He has been instrumental in founding a company, Chip Nano Imaging AS, which he co-founded at the university as a spin-off from the work that emerges from his lab, where he is also the chief scientific officer. Balpreet has currently 6 EU projects running at various stages, both intermediate and advanced, and he has 4 projects running, which are funded by the Research Council of Norway and 2 from the university itself. He has about 10 young PhD students whom he works with at the time. He even has 2 jointly guided students at IIT Delhi, and he works with postdoctoral researchers and other collaborators as well. Today, Balpreet is going to talk to us about photonic-chip based super resolution optical microscopy. Welcome, Balpreet, and the floor is now yours to make the presentation.
Thank you, Shiva, for us very generous and welcoming me to this talk series. And I'm delighted and I'm humbled to give a talk to this esteemed audience. And you have had a very nicely put forward about the work that we have done in Tromsø. So I would also at the beginning like to highlight that the work I'm going to present today is done by fantastic group of PhDs, postdocs, researcher in my lab, and also many different collaborations across the globe. So what I am presenting here is not only my individual work, is a group of people working in my group and my collaborator. And also I have a very active interest, as you will see in my talk, to work in the field of nano and microfluidics. It's one of the areas which is we have active interest, we have applications. We have just started to have some activities in Tromsø, but not had fully looked into it ourselves. So there is a very good synergy possibilities on merging our photonic chip based microscopy methods with the fluidic devices that you would make in Chennai. So the title of the talk is Photonic chip based super resolution optical microscopy. I'm in Tromsø, the Arctic University of Norway. I came here as a young postdoc. What you see on your picture here is the Tromsø island, and university is based on the northern part of the island. I thought to take this opportunity to also voice a little bit about photonics and how important photonics will be moving forward. I believe that the 19th century truly belonged to steam engines. You know, the invention of steam engines propelled industrialization and automobiles and transport. Then the 20th century was truly electronics, you know, the miniaturization of electronic circuits. And moving forward, 21st century, I believe, will slowly transit from electronics to photonics. And just to give you a glimpse of that, the market of electronics is around 1.1 trillion US dollar, and it's growing at a speed of 4 to 5%, while photonics product and its solution has already reached to around $600 billion, or $0.6 trillion market, and is even growing faster. So photonics is going to play an important role as we move forward, and that is because there are features of photon such as speed of the light, the power the photon can have it. Either you can make it work in a very constructive way, that means minimum invasion, or you can have a very high laser power to burn the metals. And the speed of photons you can't really beat that. And to put this thing in a perspective, let's assume that you want to deliver a message and you are, running a speed of Bolt. You'll probably travel 1.5 meter per second. And if you use a pigeon to deliver your message, the pigeon can fly 20 meters in a second. In more advanced time, we will either use DHL express service. A flight can bring your message 250 meters in a second, while the Apollo, traveling with the escape velocity, can bring your message 10 kilometers per second. But a single photon in an optical fiber in a 1 second can travel 200,000 kilometers. It will bring your message 5 times across the globe. No wonder we can have this Zoom meeting. I'm sitting in the Arctic and you guys very close to the equator, and in the real time is because of the high speed internet, which has backed by photonics based solutions such as optical fiber. And this is the impact which photons makes it to our life, and from the entertainment to the manufacture to the automobiles, self-driven cars. These will all be in the future based on sensors which have been triggered by photons or photonics technologies. And this is the reason when, after doing my PhD on Photonics Research Center at NTU, I looked for my career in Tromsø, where we set up a group of optical nanoscopy. In 2013, these activities have been going for last 10 years. And we are a group which are working mostly on technology development, but we are also translating its impact for different bioimaging and clinical application. All the applications here I mentioned are in a close collaboration with our partners. The method that we develop in-house are optical nanoscopy methods. They are quantitative phase microscopy methods. We have been working with trapping and also on integrated photonics. And the applications have been in past a lot in vascular biology. We are looking at women health and IVF application. Lately, we have a large project on molecular cancer and autophagy, and similarly on fishery sciences and pathology. During my talk, with the allocated 40 minutes, I will just touch some of these aspects here to talk about some of the optical nanoscopy methods and the work that we have made on chip. And I will show some examples of applications in the area of vascular biology and pathology on cancer and women health. But I will be very happy to discuss afterwards on different applications if this is interest to the audience. So the optical net microscopes is difficult to define when it was really invented. I mean, Greeks were using it, Romans were using a lens to magnify. Indian subcontinent was using single lenses, but the first patent application of using two lenses to magnify using a compound microscope dates back to 1590 when the first patent application was filed. And just to give you an idea about how long it has taken us, is good few centuries from when the microscope was developed till when we actually had a far field super resolution optical nanoscopy. It was only in last 20 years when scientist has triggered the invention of fluorescence based far field optical nanoscopy, or simply a super resolution optical microscope. So we are in the very early days of nanoscopy invention. So if you think about the problem of a common microscope, let's assume that we are interested to image a small particle of size, say 50 nanometer. Let's assume a SARS-CoV-2 virus, which is only 150 nanometer big. And we try to take an image of a virus or a particle in this case. And because this size is very small, our microscopes, even if it is had the best optics and best aberration correction, we will not be able to make the image of an individual spot smaller than 250 nanometer. So this is kind of the diffraction limit of the microscope. And when you have two such particles coming together, there is a resolution after which you can't distinguish them. You can't resolve that they are one particle or two particles. And for a very good microscopes, these are very similar to the diffraction values, which is around 250 nanometer. So basically what we are trying to say here in a simple words that you have a blur picture of everything smaller than 250 nanometer. Typically, this is even true for a very high-end microscopes. Typically, it will be around 300 to 400 nanometer that anything smaller than 400 nanometer is slightly blur in optical microscope. And as we know, the biology happens even in the nanoscale. And that has hindered the biologists over many centuries. They invented electron microscope or near field optical nanoscopy methods, which had their issues. So as I believe that seeing is believing, this is a classic example taken in our group, working with the vascular biology group, is an image on a left side of a cell membrane of liver. And this is an image which we will get with the best microscope we had. And on the right side is the same image when acquired with a super resolution microscopy or simply a nanoscopy. What you see is the same cell, which has these finite features called nanoholes of fenestrations in biology, and they are supposed to filter out the blood and take out the toxin material as the blood pumps through the liver. And these are very important for our health. And as we age, these fenestrations are lost. And to make the drugs to reverse this stuff, we need to be able to see that. Because if you have an image on the left side, you don't know whether there are nanoholes present or not, or is it aging problem or is it a resolution problem? And as it turns out in this image, this is not an aging problem, it's simply a resolution problem. And this is one just example where you need to resolve structures, very small structures, to be able to make a decision. And because of the impact of nanoscopy, the Nobel Prize was awarded to these three gentlemen in 2014 for the discovery of super resolved optical microscopy. But the field was still very new. only within like 10 to 15 years of the first experimental convincing result, the Nobel Prize was given. So that means there were a lot of challenges still to be sorted out for impact translation. And the problem was that while microscopes can image a very large area, like common day microscopes, the nanoscopies can only image a very small area, typically only 50 by 50 micrometer. And you miss the big picture. You don't know what's happening in different areas, and you cannot make a biological conclusion which is backed by statistical relevance. And they cost a lot of money. They typically cost €500,000 to 1 million. In India, it's around 4 to 5 crore rupees, and they lack multimodality. So to see what all these stuff mean, let's assume an application of pathology, in this case, a placenta pathology. What you see here is a tissue image from a placenta. And what the people are interested is to look at these nanoscale microvilli, which is only about 100 nanometer brushes around the microvilli, which are damaged in preeclampsia when babies are born before the due date. And researchers, yeah.
Sorry to interrupt, but can you tell me what you mean by multimodality in the previous slide?
So by the multimodality, I mean that, for example, you can have nanoscopes determined by a single molecule localization method, and they may not work with structured illumination microscopy. So there are different methods of implementing nanoscopy, and it's difficult to find all of them integrated into a single hardware.
Got it. Thank you.
Yeah. So I would believe that it would be best if we take the question in the end, so that we can complete this talk and move forward, if that's not a problem.
Yeah, sure. Thank you. Just a request to the audience. We have enough time for questions. Balpreet's talk is for about 35 to 40 minutes, and we have about 20 minutes for questions. Thank you. So I was back to this microvilli story. So what they were really interested is to look at the microvilli brushes, which obviously is 100 nanometer, and to give you an idea where these microvilli you can image using electron microscope. This is a tiny region down there. And if your tumor happened to be in a different part of the cell or the tissue, you have lost that space, right? Or you have to stitch a large area. And if you put them in a graph of a resolution versus a throughput, we have methods such as a microscope, standard microscope, which supports very high throughput. You can image very large areas, like you can image at high speed, doesn't really give you the resolution. On the other hand, we have solutions like electron microscope, which supports extremely high resolution, but does not support the throughput. And in between somewhere, we have this new field of optical nanoscopy. They're not quite yet there with the resolution and not quite yet there in the throughput. And what we wanted is to make an impression here to make these nanoscopes higher throughput, more affordable, more compact, and multimodality. So that was a solution I proposed 10 years back. The concept was Photonic-chip based nanoscopy. I was working as a postdoc on integrated photonics. I had background in laser beam shaping. And for me to do all the advanced beam shaping in the free space was a little bit not a preferred route. So then I started thinking about what we can make a impact here. So the synergy was that the present day solution, we used a complex microscope body to perform all free space laser beam shaping that is required for individual method. And they put the sample on a simple glass slide. I propose to converge or change that, is to take out the laser beam shaping from the microscope body and do it on a photonic chip. That means the photonic chip, which is simple optical waveguides, will act like a substrate to hold the sample and to bring the evanescence light onto the chip directly. And you can then use a simple microscope to acquire different methods. And I will talk in my talk about different methods that we can do. So that's kind of the global idea. One immediate big difference is that over the centuries, many parts of the microscope have been changed, but one part has never changed, that the objective lens has always been used to illuminate the sample and to collect the image. And we wanted to, instead of using an objective lens, we wanted to use photonic chip. And as you can see on your left image, once you couple the light inside the chip, it's just like an electronic circuits. Optical circuits can be designed based on our imagination and some simple theory of guided mode. You can parallelize that into n number and you can illuminate very large areas.
And you can do very complex beam shapings. For example, what I'm showing here in these images are 3-beam interference pattern, and people working on interferometry could realize that the difficulties of setting up beam and the common path setups to make interference pattern. While on chip, you can make it on chip. You can make the designs that you want, and you can bring all the light using a fiber coupled to the photonic chip. So the message is that if you start making your illumination patterns on chip, you can make a simple path. And for the audience about the chip itself, what this chip does is simply like an optical fiber. It guides based on total internal reflection principle, and very similar to optical fiber, except that it is now fabricated on top of a chip instead of in a free floating form. The light which is present on the surface is called the evanescence field. It decays exponentially from the surface, which is given as a exponential function in a decay length. And typically it is few hundred nanometer for the waveguides we opt now. And so also by saying that, I'm making it clear that all the methods we have developed in Tromsø so far are based on 2D methods. They are all 2D because we all only use evanescence field of the surface. So the cell is put directly on top of the chip. The evanescence surface waves illuminate that, and then you can perform beam shaping as you want. So it's not a 3D method so far in our lab. So now we move on to what we wanted to do, the concept of chip-based, and now moving towards the single molecule methods. In single molecule method, the basic idea is that if you can't separate the information in space, you separate them in a time. Let's assume this is our nanobeat, and I have 1,000 molecule on top of that nanobeats, and they are all illuminating at the same time. I don't have the resolution, but what if I can switch all of them off, and I can switch only some of them, which is separated by a diffraction limit. And because these are single molecule, I know this precisely that they are single molecule, that it has to come from a single molecule. Then I can do a simple 2-dimensional Gaussian fitting, because if I know my optical system, I know my point spread function, I can say that I don't care where this molecule is, as long as I know that this is a single molecule, I can tell with a very high precision where the center is. And then I allocate that center in my plot, and then I switch those molecules off, and I let the other molecules stochastically blink. How we do this blinking is simple by using a different chemistry, which I'm not going through into it, but this is basically a property of the fluorophores, how you can make them blink. And once you have made them blink over 1,000 or 10,000 times, you can convert a super resolved image. You can then move from this image to a super resolution image. Typically, your blinking of molecules will appear like this in the picture. So what we wanted to do is that we wanted to implement a single molecule localization microscopy on a chip. So the first task that we have to prove was, can we make these molecules blink on the surface of a waveguide? Because an optical waveguide only gives me access to the surface waves. It doesn't give me access to the guided light. And do I have a kilowatt per centimeter square intensities to make these molecules blink? So what we did was to fabricate these chip, low loss optical waveguides based on tantalum pentoxide, on silicon nitride, and different materials we have investigated. And we made a nano, well, it's not in nano, it's 150 nanometer thickness waveguide. And we checked whether the molecules can blink or not. And because of the low losses and good optical characterization, and thanks to this 150 nanometer thick waveguides, we had reached around a kilowatt per centimeter intensities, and the molecules were blinking. And once we had shown this to work, it was a matter of time that we implemented a single molecule localization microscopy on the chip. And that was our first result from our group in 2017, done by 2 very talented PhD students. We reported that we can actually make on-chip nanoscopy using a single molecule localization method, get a resolution of around 49 nanometer. And these are the images. What you see here on your left side is of a diffraction limited microscope. What you see on the right side is on the on-chip nanoscopy. Well, this is This brought us to what was the state of the art back then. But this is not what we wanted. We wanted to prove that on-chip can actually surpass the conventional technologies. So one thing was quite evident in our case, that we are not using a turf lens, because typically to make this method, people use turf lens, which comes with a very high magnification, typically 60x with 1.4 NA. And in our case, the turf light is generated by a chip. So we can simply use a low magnification objective lens. So if you invest €500,000 to buy a state-of-the-art nanoscopy from the market, your field of view is typically limited by 50 by 50 micrometer. And what we reported that using a chip, we can improve the field of view by 100x. That means in a single shot or single molecule image, which is not actually a single shot, but in a single super resolved image, instead of looking this tiny region here, you can acquire 100 times larger area. And if you zoom into this tiny region here in the green part, the resolution what you have is still sub 100 nanometer. And this was the first super resolution microscopy at high throughput. We are looking at a field of view which is in millimeter large and resolution supporting down to sub 100 nanometer. So that was our main contribution in that field. And then we moved on to see if we can implement multimodalities as the question was launched. The other method was a structured illumination microscopy. This is a different type of doing superposition microscopy. So in case of SIM, to achieve a super resolution, what you do is you illuminate your sample using interference fringes in 3 angles and 5 or 3 phase steps, and you rotate into 3 angles, you acquire 9 or 15 images, and then you send those images into an algorithm which gives you a super resolved image. Typically, this is done all in the free space. It's a very complicated setup. We bought one of these microscopes for €1 million in Tromsø ourselves, which worked fantastically. And we wanted to see if we can build that on a photonic chip. And to illuminate the sample, we needed interference fringes. So the idea was quite simple, that I can start with a single laser arm, and I can split the arm into 2 arms, and I can make them interfere. So light will be divided into 2 arms, and at the imaging area, they will be interference fringes, on-chip interference fringes. And because they will be interference evanescence light, I can simply rotate that and do phase stepping to do on-chip SIM. So the structure that we made looked like that. We used 3 pairs of waveguides. These are pseudo-colored. So like green, red, and blue, these are optical waveguides. At any given of time, the 2 of the arms will be active. For example, when green is active, I will have interference fringes on this axis. We can do phase stepping, 3 or 5 or whatever phase stepping we want, acquire the image in the camera, then I switch them off, switch then blue one on, repeat the process, and then switch on with the red one. So I can simply use an optical fiber switch, optical fiber splitters, and a waveguide chip. So there's no free space optics happening. We have the laser light going to an optical switch, going to optical splitter, and going to the photonic chip. This can work in megahertz if you want to operate in megahertz. So how does it look like? You have arm number 1 making interference fringes, arm number 2, arm number 3, and we can do phase shifting using different approaches. What we have so far delivered, we have delivered off-chip phase stepping. So it simply means that you split the light before the light goes inside the chip. You use 2 independent waveguide arms, and then by varying the phase in one of the arm, you can do the phase shifting here. It works nicely. It is an easy setup, but it's not most robust. For me, this is the best one, but most tricky one. Here is that you have an individual waveguide arm. You bring the light, you split the light into 2 arms, and you make the interference fringes at the imaging area, acquire an image, then we switch off the light in this arm, then we switch on the next arm. As you could see that these 3 different waveguides still bring the light to the same imaging area, but they are traveling different path lengths. These path lengths are all different. That means there will be a phase shift. It is rock solid, rock stable, because once you make the chip, the phase is not drifting, but it also comes with a cost that you can't change it. And in the last, we have on-chip thermo-optic where you can simply use thermo-optical devices and do a phase shifting. That was for one angle, and then I can simply repeat that into 3 angles by using 3 arms, and likewise, and I can implement on-chip sim. This was a little bit more involved work, took us a good few years, and in the end it worked out. This is an image from 3 phase steps acquired and on the chip. And the work was, we had the experimental work on chip sim actually worked. This is a conventional resolution of microscope, and this is on chip sim, which can resolve structures down to 100 nanometer. Was published last year. It was also featured in the front page of Nature Photonics. And then the last method, we haven't implemented a couple of more methods, but I'm not going into today. Last method that I'll go through is a chip-based correlative light electron microscopy. And that came because a lot of collaborator in our group, a bio collaborator, they wanted to have both the optical resolution and the electron microscope resolution. And the problems typically was that in case of correlative light electron microscope, you need to take image using an optical microscope and then bring it to the electron microscope. The problem is the correlation, how to find the same cell into 2 different regions. So that was a problem. And what we proposed is a pretty straightforward method. We suggested that whatever method we did, a sim on chip or single molecule on chip, we can simply have a global landmarking. A global landmarking simply means there will be a pillar standing next to our waveguide, and these pillars will make an imprint on the resin, which is required to embed the cell for electron microscope. So this is our waveguide running here. When you want to bring this sample to the electron microscope, you deposit a resin. And when you deposit the resin, these number systems are really like a pillar, like A1, A2, A3. When you put the resin on top of it, these number system will get imprinted on top of the chip in a single lithography step. So what you will look into that is, this is your optical image of your chip. Let's assume that you had a cell sitting at A33 here, and you can then go back and find that same cell. All we need to do is to preserve the location of that number. So this is what you get into electron microscope. Imagine we were interested to look at a cell, this one. It would have been impossible to find that cell in a large field of view. But now if I have the precise information that please go to a cell sitting at D98, then all I need is to pass that information. And this is what we did. Work done in a collaboration with EMBL in Heidelberg. Our collaborator from biology, they had a vascular biology question. We put it on a chip. We performed tons of imaging. In the end, the collaborator says that, Balpreet, we are interested on a cell P28. That's what I want the higher information of. We performed all the color imaging in the light microscopy. We prepared the sample in Tromsø, send it to Germany, from Norway to Germany. And I told to our collaborators, image the cell sitting at P28. So this was an optical measurement and done in Norway, is measurement done in Germany, P28. They had the precise information and they did a correlative electron microscopy image. That means they can figure out at very high resolution, the same cell sitting here at P28 can be imaged with ultra high electron microscope image. In this case was Fib SCM image. So that was the other work that came out from our lab recently. And then in parallel, we have done intensity fluctuation methods on chips, which I'm not covering, but can discuss in the discussion round. So basically, the take-home message is that right now we had a solution where you have to invest heavily on buying different state-of-the-art optical nanoscopes. You need to have different instruments. And what we believe is that just as when you want to change from cappuccino to latte to espresso, you're not going to buy a coffee machine. You just buy the chip. You just buy the sachet. And that's what we are thinking, that in the end, microscopes should not be complex. Microscope should be a lens and a camera, and light beam shaping can be done on chip. And for example, for us, we can put nanofluidics and microfluidics on the top to make complete incubator on the chip. And that would be a move forward. A glimpse of what applications we are doing. We are working with the Karolinska Hospital and University of Ulm Hospital in Sweden and in Norway, working on preeclampsia problems, where our hypothesis or their hypothesis is that there are issues with the microvilli on women who suffer from preeclamptic problems. We are working with a group in Unn Hospital on cardiovascular diseases or heart diseases, looking into issues with mitochondria. We are working on a cancer research activities with the Radium Hospital and also some other collaborators working on onset of the cancer and progression of the cancer. We are also interested on neurodisorders, diseases. For example, in the study, we are working on or worked with the collaboration with Cambridge University on imaging of living neuron cells. This is an example of nanofluidic devices which they fabricated and to see how does the neurons talk, and doing a large field of imaging, for example, on chip. Okay, with that, I think I had 35 minutes to 40 minutes to talk. I would like to acknowledge all the funding agencies for their support, mostly European Union projects, ERCs and Marie Curie and actions, and also Research Council of Norway, different programs from Forni to Nano 2021 and biotech projects, and University of Tromsø and DIKU for mobility grants. And I'll be happy to answer more questions now.
Yes, many thanks, Balpreet, indeed, for this very illuminating talk. You walked us through the key aspects of research in your lab, all the way beginning from the fundamentals of the Abbe limit. So it was quite a journey and gave us a bird's eye view of the entire field. The floor is now also open for our audience to pose questions and have a discussion with the speaker. We deliberately left out sufficient time in this session to make it as interactive as possible. We had a question earlier about multimodalities. So if audience wants to begin with that, we can further that discussion and also take further questions. If you have a question, please raise your hand and then you can unmute yourself and ask the question. Or if for any reason you want to just post it in the chat box, that's fine. I'll be glad to do that. Please unmute yourselves, Mr. Sudarshanan, and pose your question.
Thank you, Shiva. Yeah, my name is Sudarshan. I just wanted to introduce myself as well. I'm a new faculty in the electrical engineering department at IIT Madras, and I'm actually in the integrated photonics group. So
that's wonderful.
I'm actually glad to hear from Balpreet, and I'm actually glad that integrated photonics, we have more voices trying to promote photonics from various aspects. So, Balpreet, my question was in terms of the waveguide width and this evanescent coupling. So you have about, what, 1.5 micron wide waveguide?
No, I had the width of the waveguide was millimeter scale, 0.5 millimeter. It governs by the applications. So when we are imaging tissues, we are even using almost like a slab waveguide, 1 millimeter. The thickness of the waveguide was made intentionally very small so that we can pull up the light in the evanescence field.
Right. And what are the typical launch powers of the laser light?
So the launch power is slightly higher for a single molecule localization. Typically, we are using around 150 milliwatt to launch into the waveguide, or 200 milliwatt to launch in the waveguide. What we get into the waveguide are only a few couple, maybe less than 10 milliwatt. So our coupling losses are very large. In terms of turf imaging and the single molecule and structural illumination microscopes, our launch power is probably 50 milliwatt. So it's not much.
And what colors do you usually need? Like we saw some colors, but I don't know if they were the actual colors of the laser light.
For many experiments, they were actually, so wherever there was an actual cell image, there was a real color, except that we might have chosen the taken care of color blindness. Typically, our students do that. We typically use 488 nanometer, 561 nanometer, and 647/660 nanometer. So these are typically all the wavelengths of interest are given by the fluorophore of interest. And fluorescence microscopy use visible spectra.
And you don't worry about with these wide waveguides that you're actually exciting multiple lateral modes, right? Or transverse
Yeah,
modes.
we actually excite multiple modes. It's good that you asked this question, Sudarshan. I have not got into that. So we actually illuminate intentionally a lot of mode, and then we galvo and average them out.
Okay. And do you take care that these are T or TM? Like I'm assuming TM interacts better, right, with your...
We haven't really intentionally taken much care because the difference would have been few tenths of nanometer, would have been very difficult for us to make a difference. We just use the one which gives us a low loss because we are interested in looking at the images. But for polarization sensitive studies, that might be of interest. We did discuss with some of the PhD students, but we never floated a project. If you have an idea about where polarization-based studies imaging can be done, I'll be very happy to discuss this further. But there's there.
Yeah. Thank you. And last question, I should leave the floor.
Please go ahead. No problem.
Yeah, it's about the waveguide roughness. And like, are you currently suffering from like getting your smooth surfaces or sidewalls?
So the sidewall has not been a big problem typically because if you think about our waveguide, they're very large. You know, the sidewalls is 150 nanometer sidewall and the waveguides are 500 micrometer. It is a problem for the surface roughness. So sidewall is not a big problem. We would love to have minimum losses. Our propagation losses varies around 2 to 3 dB per centimeter for the visible range. What we suffer, and our surface roughness is around few nanometer. So it's not that bad either. But what we have faced a problem is to guide 4 or 5 nanometer. In silicon nitride platform, I mean, you are then hitting the cutoff frequency.
Right.
And then in tantalum pentoxide or titanium dioxide or aluminum oxide, and these are very interesting elements for us. And for example, what we discussed with Shiva back in India is to make these laser written silica waveguides. That would be fantastic.
Right. Cool. Thank you.
Yes, and so
thank you so much.
Wonderful. Yeah, nice to have you here, Dr. Sudarshan. Dr. Vishal has a question. Vishal, please go ahead.
Yes. Hi, Balpreet. Can you hear me?
Yes.
Yeah, you're audible, Vishal. Please post your question.
Yes. So the question was, when you use this photonics to actually, microscope image on a cell, when the cell interacts with the photon, do you think it will diffract more and then it will hamper the resolution of the photonics that you actually try to use it for this purpose?
I think I got your question. What you're saying, Vishal, is that we are using much higher K vectors in the chip, right? And when they diffract, we are diffracting with the higher spatial frequencies than what we can acquire with our objective lens.
Right. I mean, so the field width of your wave, the optical photon that is hitting on the cell, maybe actually, gets more and more losing its resolution when it interacts with the cell because of the cell and light interaction. So
Correct.
that can hamper the moment you actually inject, impose a photon onto the cell. So are you actually starting with the right width of the photon when you hit it on the cell? So that itself, I mean, that may be, pretty much a bad initial start to which may get even more diffused out to get a higher resolution, which you are looking for in the nano resolution.
Yeah. Okay. I think I now understood your question. So your assumption that the light will be scattered by the cell is right if the cell is a tissue.
Right.
Now we are talking about a cell which is 10 micrometer. Light would not see that. No problem. So if you try, I mean, maybe you're thinking more on the OCT terms where you're trying to use laser to image inside the tissue. Yeah, light scattering happens, and then it's a problem. Not for imaging a cell which is almost transparent, doesn't really change the momentum of the photons.
Right. But when the light is, what is the width of the light that is hitting on the cell? I mean, is it the intended width is actually much lower than the cell width, or the intended width of the light that is coming, that is being focused initially is more than the cell width?
Yeah, so by the width of the light, you mean by the width of the waveguide, right, Vishal?
Yeah,
so in this case, so the width of the waveguide must be bigger than of the cell that you are interested, right? So we are using a width of waveguide, which is say 500 micrometer, and the cell is like 20 micrometer, so that I can image hundreds of cells at a time. And then when we have the modes, these modes of the waveguides are very small, and then we average them out, and we have hundreds of modes going through it.
So if that's the case, so the last question, I mean, I think I see what you're, we are on the same page on, in terms of the interaction of the light and the cell. So if you reduce the light for width, the diffraction, the width of the waveguide from 500 microns to smaller and smaller, I mean, so how does the cell resolution get affected? I mean, does it serve the purpose of nanoscope imaging, or is 500 the optimum choice to start off with, 500 microns?
We can make any structure size. That's not a problem. So the width of
the size of the the size of it should be in the hundreds of microns to start off, the waveguide size, to actually get into the super resolution mode, or can it be even micron size to actually get into the nanoscope? Because you're talking about almost from micron to nano, almost you need to reduce it by another 100 times. So if you're already starting half a mm, then you're actually talking about 10 power 6 times to a million times to reduce it to do a nano imaging.
Yeah, so maybe you have misunderstood the concept. I can explain you a bit here. So the width of the waveguide has nothing to do with the resolution. Your resolution of your microscope is given by lambda by 2 Na. Lambda is a wavelength of the light you illuminate. Na is the effective numerical aperture of the objective lens. So your resolution has nothing to do with the waveguide width. What we are using.
I'm talking about the resolution getting affected because of the cell interaction with the waveguide.
Yeah, so
you have the cell to actually capture.
Correct. So resolution is not affected by the cell simply because it's a fluorescence method, right? So what we are seeing is a fluorescence light, and fluorescence light getting through a cell which is only a few tenths of micrometer will not have any aberration problem, because your resolution will be spoiled because of the aberration issue, right? What you're asking is how does the aberration from the cell distort the resolution? And answer is not significant. Not significant to measure it.
I understand. I mean, coming from the top that you have presented, you had multiple cells or molecules where you are able to, distinguish that it was resolved properly. But when you go into finer and finer, let's say a couple of particles or a couple of cells alone, which are of, when you isolate them and then want to actually focus them and capture the image of that, is it possible with ensuring that you can take care of the interaction between the photon and the cell? I mean, that's the last question.
I don't get your question, Vishal. Are you thinking about a 3D structure cell, or are you looking into a flat structure cell?
Flat, 2D.
2D should not be a problem, right? Then I don't get this. What is your problem with this question? So you have a waveguide, you illuminate the light on the waveguide, you have a cell on top of it, you put an objective lens to image it, right? Your light illuminate in this path, your scattered light is captured in the upward path, right?
I'm talking about the fineness of the resolution. Right now you have a 500 micron where you can capture a 20 micron. Now, when you go to smaller and smaller particle cell, let's say you got probably around 100 nanometer. What should be the
resolution? So maybe I can guide you through. I think the interpretation of the resolution and the width of the waveguide are decoupled, Vishal. So they are two different things. So the resolution is given by your signal to background noise of your fluorescence signal, which is given by the intensity of the fluorophore brightness in terms of single molecule localization. And how precise you can localize that molecule gives the resolution. It has nothing to do with the waveguide, it has nothing to do with the properties of the chip.
I know. Sorry. So just to
I'll come back to your point. So if you have a molecule sitting inside the cell, and let's assume the cell is 10 micrometer wide, and it's only illuminated by a sheet of light. So wherever these molecules are, it has to be within the field of view of the chip to get excited. So if it is not within the evanescence field, it will not get excited, and because it has not get excited, you will not see it.
Yeah. Okay. May I just, if you don't mind, may I interrupt the discussion, Vishal? We can come back to this. Can I take another question? Yes, Vishal.
Shiva, just last thing. I think I got what Balpreet was saying. Just last thing, when you actually see with this perspective, I got what you're saying, that you're interested that whether the image has been captured, then how do you distinguish what is there inside the cell that you're looking for?
So that's
When
fluorescence.
you actually,
That's fluorescence.
okay, through the fluorescence. Okay.
Yeah. I
Thank
mean,
you.
that is, everything is fluorescence, right? Otherwise we are blind.
Okay, sure.
Yeah,
Thank
sure. I can take the last question and then I
Yeah,
have a
sure,
meeting.
indeed. Yeah, in the interest of you have an engagement, Vishal, but we'll definitely invite also Balpreet for a great engagement with our group. We'll
Of
have
course.
time, I
I hope
promise
it's
you.
not too cold right now, Balpreet, that even at 12 and I
Yeah.
guess it's a February 5th, so yeah,
Yes,
February 16th.
absolutely. Yeah, there is
I
a
saw that it's like 5 degrees outside. So I hope it's
yes,
not too
yes.
cold.
Yes, Vishal. Thank you. There's a question from Andre here. He asks, would there be a way to extend near field excitation of the chip to the far field in one or another way using microlens arrays at the surface or other means?
Good to see you, Andre. I hope everything is well with you. I would have loved to do that, Andre, myself, as you know, as much as you know, I would love to do that. And you are bang on. I would love to use micro lens array. So we have solved one part of the problem. We can illuminate millimeter area, but we can't collect millimeter field of view. So I would have personally loved to be able to illuminate 1 millimeter and collect 1 millimeter. That is kind of the microlens array for the collection light path. And then other stuff could be of interest of, as you have said here, is the surface roughnesses, and other stuff can also be playing important. We have started looking into ways of enhancing the we are in ways of enhancing the spatial frequencies that can be carried by the waveguide. Here, the plasmon or dielectric plasmons could be of interest.
Yeah, Andre, if you have any further thing to ask, you could even unmute yourself and pose the question. But otherwise, thank you, says Andre Schmirov from Germany, very much. Thank you, Andre, for joining us today, as also with all other folks here and on the YouTube stream. Well, I guess a talk like this also spawns of many ideas and further discussions. So this really means that we have more reasons to engage with Balpreet at various levels and ways. So we will certainly do that, and we will invite you, Balpreet, for maybe further interactions. Maybe a series of lectures would actually be a perfect way to do it, maybe staggered, and they can collect this, have an audience that comes back to us again and again. I really look forward to putting together such an event. Of course, Balpreet has a commitment waiting for him in a few minutes from now. So it's only appropriate that I thank Balpreet now for this very stimulating and, as I said, illuminating talk by myself. And so thank you so much, Balpreet. I hope this is not just the conclusion of this lecture, but the beginning of an enduring interaction with our group here at IIT Madras. And as you also saw, colleagues are working on various aspects of photonics, as you also probably realized during your visit, and certainly there is a lot more to come in the near future. I will close with just leaving a little bit of time for Pallab to say something that he would like to. He puts together this very nice talk series along with our very enterprising student, Mr. Deshu Harsha, who does all the background job very silently, very efficiently, but also always perfectly. So thank you, Harsha, as well. And Pallab, it's now.
Thank you, Siva, for this talk and for this organizations of the talk, and Harsha definitely, and our team, and Balpreet. It's a nice talk. It's a wonderful talk, and as Siva said, this is the starting of our discussion. So we will contact you again, and we will ask for further discussions, a detailed discussion, maybe more informal discussions, and we will look forward for that. So
Thank
with
you,
that,
Prabha,
thank you,
Shiva, and I hinted, Shiva, that there's a huge push on collaboration between India and Norway. So there have been a series of funding calls aiming towards the mobility, and one of the project that I had with IIT Delhi, and I think there's a huge call now opening up with the Norway and India. Would be nice for us to merge on this photonics. I see that you have integrated photonics activities and also nanofluidics and microfluidics are of much of interest to our group.
Sure.
I have to rush now for a meeting in
Yes, Balpreet.
Yeah.
So
Thank you,
very much.
Balpreet. We'll
Safe.
talk later. Yeah.
Thank you,
Bye.
Balpreet. We'll catch you again and we'll have a focused event. Thank you so much.
Thank
Bye.
you.
And thanks to all the audience. And if you all permit us, we will close the session here, unless there is a coffee table discussion that any of us want to engage in.
Yeah,
Thanks,
I also
everybody.
have to leave, Siva. So thank you.
Thank you so
Yeah.
much. Thank you, Pallav and everybody.
Thank you.
Thank you, Harsha. I think we can close.